So could you tell us a bit more about how you use DNA within your research?
Well, I work on a virus called Merkel cell Papilloma virus. It causes a very aggressive type of skin cancer, so it's important that we know how it does it and how to stop it. Now, working with DNA is an important part of my work. While I don't specifically investigate DNA as such, I use it as a tool. So, for example, I have a particular protein of interest that my virus produces. So I might want to visualize this protein so that I can see if it has any effect on cells. And one way to do this is to add a fluorescent tag. Now, a fluorescent tag is just another protein attached to my protein of interest, and it emits light under a microscope. So one you may have heard of is called the green fluorescent protein, or GFP. Once I've made my new DNA construct, I want to put it into cells that I work with. And different scientists use different types of cells, or cell lines, depending on the research. So because I work with a virus that causes a type of skin cancer, I use a few different types of skin cell lines. So how do I get DNA into my cells? Well, it's a process called transfection. And DNA is a negatively charged molecule, and it needs to go through the cell membrane, which also has negatively charged molecules called phosphates in it. So transfection reagents are different positively charged compounds that bind DNA and help it move across into the cell. So the gene that the DNA contains can then be expressed in the cell and visualized under a microscope with the help of the fluorescent tag. So in the end, all this hard work can lead to images like this.